I try to imagine a fellow smarter than myself, and then I try to think, what would he do?
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. I'm your host, Jesse Lawler, here with episode number 63 of this podcast dedicated to helping you better your own brain by any and all means at your disposal. This is an episode that I actually had to get military clearance simply to get on the phone with our guest, which, if nothing else, should give you an indication of how excited I was to get on the phone, because, the harder it is to get on the phone with somebody, generally, the more you assume it's going to be awesome. And in this case, I was not disappointed. We're going to be interviewing Dr. Adam Russell, who is the program director of a program called SHARP at a government agency called IARPA, which stands for the Intelligence Advanced Research Projects Activity, and is part of the Office of the Director of National Intelligence. And they've got a cool sci-fi looking logo. Basically, the whole organization, including their website, it looks pretty much like one of those awesome government agencies that they show in futuristic movies that you're like, wow, that'd be a cool place to work. And anyway, we're going to be talking with Dr. Russell in just a few minutes about what the SHARP program is, what they're attempting to do there, teams of scientists that they have working on quite literally making already smart people smarter. So it's right up our alley. If you hang around until the end of this episode, I'm going to tell you about an easy way to make yourself more creative, and significantly more creative. This isn't like a 2% difference or something like that. This is a pretty major thing without any sort of weird behaviors, without any sort of pharmaceuticals, without any sort of anything that you'll find overly objectionable. And for right now, I'll leave it at that. So let's kick things off with This Week in Neuroscience.
Smart Drug Smarts. This Week in Neuroscience.
So published recently, and we actually have the link to the full study, which is downloadable for free, is one of the largest studies ever done on the cognitive effects from alcohol use. And this is interesting because it's broken down by sex, and apparently there's a lot of differences between the way that male and female brains handle alcohol, some of which might be because the sexes actually metabolize alcohol differently. So it's a large study, a lot of findings. Some of them were more statistically significant than others, but probably the most interesting and statistically significant takeaway was that for men in particular, heavy drinking was found to significantly reduce cognitive function. This was a 10-year-long study, and the heavy drinking men in the group, who were those that were drinking, I believe, 36 grams of alcohol a day, which is equivalent to about 1 fluid ounce, gained a few extra years of brain age in addition to the 10 years of normal brain wear and tear that would have been expected just from the passage of time. Heavy drinking men picked up 1.5 extra years of brain age, equivalent to loss of function in executive function, and 5.7 extra years of brain age in memory. But interestingly, this was not seen in women. It's unclear why this is. A study conducted in 2001 by the University of California, Davis showed that estrogen actually acts as a guardian of brain cell health, and the higher levels of estrogen are linked to reduced risk of Alzheimer's disease. It's possible, although speculative, that estrogen may also act as a hormonal shield against alcohol toxicity. Now, this isn't to say that just because you're a woman, you should go out heavy drinking all the time. This study was only about the effects on people's brains, and there's lots of other reasons to drink in moderation, not the least of which is liver health. And there were also some strange and fairly counterintuitive findings in this study, although they didn't reach the same level of statistical significance where the researchers felt really confident in them. But women who totally abstained from alcohol actually showed their brains aging at a faster rate than women who drank low to moderate amounts of alcohol. But there could be some confounding factors in the self-selected group of people that didn't drink at all. We'll put the link to this research up on the website, so check it out. Happy to report I picked up another 5-star review on iTunes from the listener Interested In, who said, "So many things are hated or loved that nobody takes a look at from a neutral point of view, making it hard to get unbiased information. Smart Drug Smarts, though, nails it." Thank you so very, very much. And actually, this was a super busy week as far as interaction and communication with the Smart Drug Smarts listenership, because, as I've mentioned, we're doing the Water Fast week is coming up in less than 2 weeks now. February 16th is when we're kicking that off. And a couple days ago, we opened the door to a little private forum for the people that are going to be participating in the Water Fast. We're all kind of meeting one another and figuring out how and what we're going to be doing for that week. Should be a really, really interesting experience. I'm at this point really confident that the upside of meeting all the people that have opted into this is going to outweigh the downside of being really, really hungry for a while. And apparently having kind of like flu-like symptoms for a couple of days in the middle is the normal thing to expect as your body downshifts into ketosis. But needless to say, there will almost for certain be an episode in a week or two all about the water fast. Just it's going to be an oddball experience worth filling everybody in on. But yeah, if you're listening to this and it's, let's say, anytime up to like February 10th, and you'd like to get in on the water fast yourself, we're going to be doing it from the 16th to the 22nd of February. And you can just drop an email to jesse@smartdrugsmarts.com if it's something that at the 11th hour you want to get involved in. Well, I've got a quick callback from a couple episodes ago when we did the marijuana episode. A friend of mine reached out and reminded me about an incident from, this is almost 2 decades ago now. But I was never a pot person, but I grew up in Oregon, and I was probably one of the only people in my university that was not a pot person. But anyway, a friend of mine had a job where he needed to get a urine test, and he smoked pot on a frequent enough basis where he knew that he wasn't going to be able to handle that with his urine. But he knew that I wasn't a pot person, so he asked me if he could have some of my urine, which I said yes. And so I gave my friend a thermos with more than enough urine for his purposes, which I always thought this was hilarious. But happy to help a friend out. So I gave him a clean bag of urine. That's not an oxymoron. And he snuck that into the testing facility. Now at this point, the urine was probably at least a day or so old. I don't remember how much lag time there was, but he knew that he needed to provide urine that was at a reasonable body temperature. He might have been a pothead, but he wasn't stupid. So I'm not sure exactly how the microwave got done. I mean, he was a pretty resourceful guy. So he might have had a microwave in his car hooked up to his car battery or something like that. But either way, he microwaved my urine before sneaking it in there. And so he, pretended to go in the bathroom, poured the thermos full of urine or whatever into the urine cup and gave it to him for the screening like, nothing was nothing. And he got a phone call back a couple days later that he needed to come in for another urine screening because there was a problem with the urine. He played dumb. He's like, "What's the problem?" I said, "Well, your urine was 109 degrees. Do you have any idea how this happened?" And he was like, "No, that's really strange. Maybe I had a fever." Anyway, so he had to take another urine test a couple weeks later. I think he actually managed to get that one to work. I don't know exactly how he passed it the next time. I know it did not involve any of my urine, but that's my historical pot story.
Smart Drug Smarts.
So Dr. Adam Russell, who we're about to speak with, is the program director for SHARP, which is a clever acronym for Strengthening Human Adaptive Reasoning and Problem Solving. And as we'll talk about, is specifically looking to increase fluid intelligence, which I think we haven't talked about the definition of fluid intelligence for a while. I think it was the episode on the book Smarter, maybe about in the 30s of the Smart Drug Smarts podcast. But the short version is that fluid intelligence is really kind of what we think of as mental agility, the ability to quickly play with ideas, look at them from different angles, sort of rearrange, see how things link together. It's not tied to information. It's not, do you know X? It's if you're given ideas X, Y, and Z, can you roll these around in your head and find interesting ways that they intersect? And the SHARP program is a 3.5-year study divided into 2 phases. The first phase is going on now, looking at a variety of different interventions that could be used to increase fluid intelligence in already high-functioning people. So the overt goal is to make the people that work in the intelligence department of the U.S. national security apparatus even smarter, but obviously there would be tons of follow-on benefits for presumably the entire species if we can figure out how to crack this particular chestnut. So let's jump in and hear what Dr. Russell has to say. First of all, can you give a quick rundown of what IARPA is? I think most people will probably have heard of DARPA, but IARPA was a new one for me, acronym-wise.
Right. Yeah, no, that's, it's actually, it's becoming less common to run into that, but understandable. So IARPA is actually modeled on the DARPA model of what's called high-risk, high-payoff research. AR stands for advanced research, and then the P stands for projects, right? So the idea is that we actually have projects that are stood up and run by program managers here at IARPA, all of whom are on term assignments, by the way. So you come in, you work your projects, and then you leave. And that's I think it's a strong and important business model, particularly in the government. And the idea is that those projects come with shelf lives. So anywhere from 3 to 5 years, depending on the actual research itself. And the idea is, you're kicking it off, you manage it to the end, what the logical end is, or the technical end, however that's defined. And then that project essentially goes away with the results of it being captured, archived, and in some cases transitioned, although that's not always the case with IARPA programs, largely because we do what's called, and the distinction is important, fundamental research. A lot of people mistake fundamental for basic research, and those aren't quite the same things. Fundamental is that high risk aspect of it may not be technically possible to do this, But if it were possible, it could make a huge impact on how things are done by the intelligence community and probably society on a larger scale, such as with SHARP, for example. So if SHARP could actually improve fluid intelligence in high-performing adults, the implications from a societal level could be very large and beneficial for all of us. So IARPA was stood up 2007, formally stood up, as with any organization that just sort of has been stood up. It takes a while to get your legs, but we are actually designed to be outward-facing. We have a website, which is sort of unique sometimes among the intelligence community. We have a Twitter feed. So we are very much reaching out to people and performers who might not have considered working with the intelligence community previously or been aware of funding opportunities there. And I'm a social anthropologist by training. So it's a really it's been a unique and I think really positive experience for me to be able to reach out to behavioral and social science research communities in a lot of cases to say, hey, here are really big problems. I think the world would be better off if we actually could address.
For this high risk, high reward sorts of projects that you're undertaking, do they have sort of a percentage in mind when they're granting funding to these projects?
No, that's a great question because it can seem a bit of a black box. The way we solicit proposals is through something called the broad agency announcement. It's not the most gripping reading in some cases, as sort of government legalese has to be. But the reason I'm sending it to you is because we have what are called the Heilmeier questions. They're there to help us evaluate different proposals. We actually have the criteria spelled out in the BAA, or the broad agency announcement, and these criteria are applied to any proposal using what's called a source selection evaluation board. So this is a composite of subject matter experts internal to the government. And they are, it's actually a very intense process and extremely rigorous, by the way. Until you've actually been on one, you sort of wave your hand a little bit and think like, well, they're throwing the papers down the stairs and whatever lands to the bottom, they fund. On the contrary, it's really intense, many days in some cases, and you're evaluating all these proposals based on scientific or technical merit. So, does it make sense what they're trying to do? Are they proposing cold fusion or something? The work plan. So, are they actually going to be able to get to where they say they're going to be? It's all well and good to say you're going to enhance fluid intelligence, but it's going to take a lot of people and a lot of work to get to these milestones. Experience and expertise to have the facilities. If you're going to run something out of your basement, that may be appropriate for some programs, less so for others. And then there's actually the relevance to IARPA as well. So a lot of people come in with, there are a lot of cool ideas out there, and we can't fund all of them. And so I think the idea from the relevance aspect is where should IARPA be compared to, say, an NSF, where that's much more sort of basic research in some cases.
So for SHARP in particular, the program that you're heading up, that's one of the ones that made it through all these hoops successfully and actually made it down to the bottom of the stairs.
That's right. Yeah, I fell down the stairs successfully. That's how I think of myself professionally. I was always voted most likely to be hit by my own train of thought. So the way the program itself works is again, program managers will develop these ideas. They'll spend a lot of time, a reasonable amount of time actually, doing sort of market research. So one of the things IARPA does regularly, and you can see it again on our website, are these things called RFIs, or requests for information. And that's a way of sort of testing the waters. And it says, okay, here's an area. We can use one that just came out recently called Fast Forward. Here's an area that has to do with sort of wearable sensors. We know they're exploding in numbers. We know they're increasingly ubiquitous, but it's not clear what people really can pick up with it. It's not clear if they're useful as a research sensor, for example. So these are questions we're asking a much larger community, and then they'll respond, if they're so willing, with these sort of very short white papers. And among other mechanisms, that allows a program manager to look and say, okay, now is not the right time. The question's either too hard or there isn't a sufficient body of research to go, or expertise to address it. Or, in fact, this is the right time. Like things have changed sufficiently to think that we can do something now we couldn't do, say, 3 or 4 years ago. And then that program has been put together and essentially pitched to the higher ups. And again, every level there's these subject matter experts, and that's where you're addressing the Heilmeier questions. What are you trying to do? How's it done today? And what are the current limitations? Why do you think you could be successful, right? If it's so important, why hasn't somebody else done it? Who cares? I mean, so congratulations, you can do it. Does that make a difference? And then 5, which is some people look at as a sort of afterthought, but it's actually the really hard part, especially when you're studying humans, is how are you going to measure your progress? How are you going to know for a fact that you're getting somewhere technically, and how much is it going to cost?
Yeah, and especially some of these things on a relatively short time horizon, like you have to measure something like, are the people actually getting smarter on the results of these interventions, and will that last long term? You don't even necessarily have a long term to do those studies.
Yeah, it can be a real challenge. In fact, I'm pretty proud of the performers at least in phase 1A. Testing a lot of people with pretty rigorous metrics, moving into the next phase, large numbers of subjects in a 12-month period. That's difficult to do. The nice thing about this is, if there's a project worth funding in IARPA, generally the idea is it's worth doing right. So unless there's funding in place for it and the priorities are appropriate and it's done carefully, it probably won't get funded, because the realization is that funding something halfway can almost be worse than not funding it at all. You end up with these results that may or may not be true, and especially with human subject research, you never want to accelerate that past an appropriate research timeline. So all of our human subject research programs go through appropriate civil liberties and privacy office review. They go through the human subject research protection process, IRBs at the local, in some cases government level. So you have to build that into the program. So not only is it now you have to test a lot of people in 12 months, but you have to test a lot of people only when everything is in place, and that can take a while too. So it's ambitious, to say the least.
So can you dive in a little bit with the details of what you're trying to achieve with SHARP?
Intelligence research, and I mean it with a small I. Spearman a long time ago identified that there's this thing called G, right? General mental ability. And so the appearance was that if you do well on one test, you tend to do well on a lot of other tests that test mental ability. And he called that G. Actually, it was referred to as intelligence, and he immediately sort of regretted that, because he knew that was going to come with sort of value-laden. But since then, psychometrics have generally broken it down into crystallized intelligence, or GC, and fluid intelligence. And I need to pronounce that carefully, because a lot of people hear food intelligence, they get very excited. The easiest way to improve crystallized intelligence, right, is to send somebody to school, essentially, or put them on the job. Fluid intelligence has traditionally been sort of considered, kind of a fixed ability, where it's what you do with what you know to resolve or deal with novel, complex, and ambiguous problems. So challenges where you can't actually learn the answer in advance and apply it accordingly. And again, so that was sort of considered and tested as a sort of fixed capability. Well, 2008, a couple of studies came out where they claimed that they were able to do what's called far transfer. So they could train on working memory, and they would see improvements on working memory, which makes sense from a practice perspective. But they were actually seeing transfer to other measures of fluid intelligence, which it was roughly the equivalent of Roger Bannister breaking the 4-minute mile. Up to that point, people, there's no way you could do this. And now it appears that you could. Got a lot of scrutiny, understandably, because that's, God bless science. And since then, the results have been equivocal. I mean, sometimes you see it replicated, sometimes you don't. There's been a lot of claims in areas like transcranial direct current stimulation, the brain training. Lumosity certainly wants to give people the appearance or the impression that they can do this. But it's all sort of been lots of smoke and not a whole lot of fire. And a lot of that is due to the sort of methodological concerns, small numbers, in some cases using only one or two fluid intelligence measures, which is, if you're trying to improve a construct, you should be able to see that across a number of different tests. It shouldn't just be one test. Randy Engel at Georgia Tech has a very good description of this. When Yaki and all came out with their first working memory studies, he was saying, well, if you really do want to test this, these are the approaches you need to adopt. And then himself and some other folks did actually test it, and they didn't replicate. So, long story short, there's something there. It's not clear what it is, but if it were possible, like I said, the implications could be pretty big, given the amount of what we call the variance of performance is explained in the real world through something like fluid intelligence. It seems to be really important for complex environments, and the more complex, the more important it is. So, if you can bump that up in people, like I said, I mean, the implications for being able to adaptively reason and problem solve in these sort of novel, ambiguous environments could be improved, and that's great for everybody, really. It certainly could be expected to improve analysis, but also, from a scientific perspective, a rising tide, floats all boats, and so. From a social perspective, that would be enormous if it's possible. So that's what we're doing. Yeah. So the SHARP program is looking at are there sort of safe, reliable, rigorous interventions that can actually improve fluid intelligence in high-performing adults. And we have a number of different performers who are taking different approaches as to trying to do that. I don't want to bore you, but I could talk a little bit about that.
No, bore us by all means. That's sort of the next reasonable question, I guess, is that there are a lot of different ways you could approach this problem. From, you mentioned transcranial direct current stimulation. There's sort of a host of other things, whether it's pharmaceutical approaches or implantable technology. What are some of the roads you're going down?
Right. So essentially, when we put out the original request for information, that sort of market research that I mentioned, we got a lot of responses. I mean, this is sort of what told us it was going to be an interesting program. And they covered the full gamut. There were people proposing, all those things, and then some. My particular favorite was naps, which unfortunately we're not conducting research on naps here yet. Although, who knows? Maybe cross my fingers. In the future, I'll be hailed as the guy who brought naps to the government. And again, we say anti-naps is kind of anti-science. It's clearly improves people's performance.
Yeah, I think the Stanford Sleep Research Center. It's like they've got paper after paper after paper proving the benefits of napping and sleep.
Yeah, it's such an easy give too. And what's nice about it is in this area is, as far as we can tell, there really aren't many side effects to napping. But other than the social interpretation that that guy's lazy, which if you're doing a 20-minute power nap, I want to be on your team.
You just need those glasses that have the eye on the outside of the glasses, so it looks like you're still awake.
Yeah, I gotta get a pair of those. That's a good intervention. So actually, the teams that were funded under SHARP are taking sort of multi-intervention or multi-combinations of interventions, and the understanding that if fluid intelligence is this core cognitive capability, adaptive reasoning, problem solving, then one simple approach probably is not going to be enough to move the needle. So one of the teams was looking at combination of mindfulness plus, let me say all the teams are exploring brain training, but it's not brain training in the sense of Lumosity. These are games that were developed from first principles all the way up, meant to train specific executive functions, inhibiting, updating your information, shifting, et cetera, and have, I would argue some strong science behind them as to why it might work. And then in addition to the brain training, which differ across the teams, they also a combination of things like mindfulness. We do have transcranial stimulation of different kinds. So there's direct current stimulation is the most common one. There's also like random noise stimulation, and then we also have exercise, which sort of causes people to pause a little bit. Except that it's if you look at the effect sizes and the influence and the results, it's pretty convincing. So, on its own, will it actually enhance fluid intelligence? Don't know. But, what you're probably doing with exercise is leveraging your sort of endogenous pharmacy, right? You're creating an environment for the brain to be in a good state to adapt to this training and ideally show that far transfer. The brain is housed in the body, so I think there's something to this.
Yeah, and I mean we know the opposite's true. We know that as your body degrades, your brain is sort of going to degrade right along with it. So it certainly would hold that the opposite should be true.
No, I think that's right. I think that's and we'll talk about how we're measuring success, which I think is a really important part of the SHARP program too. But we're asking for convergent validity. So we're also asking people to measure the neurobiology of the subjects or the participants who are undergoing this to help explain. If you're improving something that this important, you should be able to demonstrate that not just on psychometric or behavioral batteries, but there should be identifiable changes and sort of the neural correlates, the structure and function of the brain that explains or correlates with those things. And we live in an era now where fMRI is not that difficult to do. Other aspects of neuroimaging, EEG, et cetera. So in some cases, it's just a matter of also measuring things like VO2 max. So if you're more efficient at transferring oxygen and other substrates to the brain, it's not impossible to think that actually would improve your ability to adaptively reason and problem solve. So these are all hypotheses that are being tested in the program.
What does a day in the lab look like for you? I mean, you're mentioning all these different teams. Is this like teams sort of competing with one another in a good-natured fashion, trying to literally outsmart one another? What's actually going on in there?
So I don't conduct any of the research myself as a program manager, right? So my job is to try to ideally put a good program in place and then get the best people and the best minds to work on that problem, which is that also has been a fantastic experience being at IARPA. Is you get to work with rock stars. One of the performers performers, for example, when it comes to the exercise intervention, is the University of Illinois Urbana-Champaign with Art Kramer and Chuck Hillman and Aaron Barbet. And those guys have been working in this area for years and are really excited to have the opportunity to ask this question of, again, sort of healthy, high-performing adults. It's one thing to be funded on our NIH to deal with, sort of, as you pointed out, the degrading brain, the aging brain, et cetera, all of which are very important, by the way. I'm not suggesting in any way, shape, or form that sharp research should supplant that, but it is a different question. So that's what we're doing, actually, moving into the next phase, that there really is kind of an impressive sense of collaboration even among the teams. So they're not sharing any interventions in particular. They're not, sort of swapping stuff. That we need them to remain independent, but they are sharing data and information where it makes sense and leveraging each other's strengths. And I think, while every team wants to be successful in their own right, I think, as you mentioned before, the implications of this and the general, goodness that could come out of this, I think, motivates all the teams to be in this sort of this spirit of collaboration rather than total competition. And we've had similar experiences with other programs here where our performers have actually published null results, and I think importantly so. Again, after the interview, I'd be happy to send you a link to a PLOS One article that was recently published by Air Force Research Lab, in which they call into question, I think, a lot of the real excitement around oxytocin as the moral molecule or the trust hormone.
Yeah, oxytocin's got so many people saying so much good stuff about it. It's it'll be a little sad if that doesn't turn out to be true.
Well, I think it's I always remind my performers of what's called Orgel's third law, right? So biology is always more complicated than you can imagine, even after accounting for Orgel's third law. So it's never a straight story, and at the very least, again, finding a null result is important because it tells us that, okay, it's a little bit more complicated than we thought. And I'm not saying oxytocin is not an important neuropeptide at all. I am saying that this idea of oxytocin is a trust molecule does not seem to be supported, at least when you test it in people using a valid assay. And that's sort of the concern. And again, this is tangential to the main topic, but it does reflect this idea of having to use reliable measures. So if you're using a measure that you don't have, that you haven't actually validated, you may get these results that may not be true. They may be a function of measurement error. And in some cases, it appears that some of the early oxytocin research may have been like that. But they're getting this interference that seems to lead to this conclusion that oxytocin is responsible for something that it may or may not be. And we're actually really concerned not to, or at least to tackle that challenge as well with SHARP. And so one of the things we're doing within this phase coming up is we have a really rigorous, highly reliably tested, what we call T&E, or test and evaluation, fluid intelligence battery test. So this was developed in combination with Educational Testing System, or ETS, a host of different measures that all which load on fluid intelligence. And they were, we've got the psychometrics. We've tested on, hundreds of folks, knowing kind of what the ceiling effects are, does in fact, because one of the challenges with these measures is if you're really smart, sometimes you can top out quickly on these measures. So if we make you smarter, there's no way we could actually pick that up. So our performers are generally aware of what's coming, but they don't have access to the actual measures. So we're a pre and a post test that are essentially invisible to them. So if and this is not because we don't trust them, it's because, as Richard Feynman says, science is a way of setting up a system to prevent you from fooling yourself, right? And bearing in mind that you are the easiest person to fool. So everybody wants to believe this is possible. And so we have to be very careful not to fool ourselves. And I think this is the right way to do it. So we have highly reliable measures, and if performers move the needle on these measures, I probably one of the more committed skeptics, will acknowledge that this is doable. There's something there.
So what does the theoretical goalposts look like for the SHARP project? What would be a definitive success from your perspective?
Well, the way we set it out in the program, and we're very clear about these metrics, which you can again get from the broad agency announcement, is we're looking for quantitative improvements between different groups. So if you were given an intervention of a sort and the other group is given an active control, there should be a demonstrable improvement between those two groups. And the hard part of doing people is that they're inherently fuzzy, but this has to be sort of quantitative enough and based on common sense that you can interpret the results. So I think with the measures we have enough there. So that to me would be success, at least as we've defined it, programmatically. And it gets pretty ambitious. By the end of the program, we're looking for almost a full standard deviation improvement from one intervention group compared to the active control. And the challenge there is the active control. So a lot of research has been done with an intervention group and then, for lack of a better term, a placebo. So you sign up and then you just stay at home for 6 weeks. And when we compare our intervention to those groups, we have active control. So we have the performers are all having active controls where they're using interventions that are actually in themselves showing some improvement. So that's the nice thing about this. This is real world. So the challenge is how do you come up with a battery that remains something that tests fluid intelligence, and that's what we've done. So by using multiple measures, many of them novel that people haven't seen before. So, the improvements will be done in terms of those scores. So some of the tests, will be 10 to 20 points. Others will be 30 to 40. It depends on the degree or how they're scored. But you're still looking for a pretty significant bump up. That's by the way, at the end of the phase. So to be realistic about this, it's a lot to ask people to get out, go out of the chute and enhance fluid intelligence immediately by standard deviation. That's just not going to happen. So it's a staged progression. But the notion is that by getting to the phase 2, you demonstrate this is possible over large numbers of participants, but we're talking hundreds of participants. In this phase, almost 1,000 participants will be involved in one group or another under the performers.
And this is a 2-phase study, right? We haven't actually talked about that yet, but over the course of 3.5 years, it's basically half and half, phase 1, phase 2?
Essentially, yeah. So phase 1A was sort of proof of principle, give people time to figure out what works and what doesn't, as well as to get a sense of their, what they call the battle rhythm. So it's really, really tough to measure this number of participants over this amount of time. In some cases with, an active control, it can be kind of difficult. You're asking people to exercise. Americans are not famous for this. So you will have some participant attrition. You'll have people who just don't come back for whatever reason. And you have to calculate that as well. So phase 1A was kind of, get your feet wet and figure it out, show some neurobiological demonstration that something's changing, as well as give us a sense that, indeed this is safe. Phase 1B, where we are right now, is no kidding, show effectiveness against this T&E battery. Then the second phase, if we hit those metrics, and it's considered credible to a team of experts, the second phase is all about tailoring the approaches for individuals. It's individual variability has been a real bugbear in a lot of research. What works for you may not work for me, or may not work as well for me. If I already have a sky-high VO2 max, what's the feasibility of improving that? Whereas I may respond really well to transcranial stimulation or some other intervention. And the idea is that by collecting these neurobiological data in the first phase, we will be able to develop predictive models. So you, Jesse, based on these aspects about yourself and how you test, this is the intervention for you, or the combination of interventions for you. And I'm really excited about that, because I think that that's the appropriate way to go with this. It leverages your individual strengths, address where you can be stronger, and that's the reason why we believe that we could actually get an even larger effect size. When does phase 1B draw to a conclusion? We are scheduled to come to conclusion December. So we just kicked off phase 1B this month. They're in the process of getting IARPA approval, human subject research protection, the oversight, getting things in order. The facility stood up again to essentially replicate phase 1A with improvements, lessons learned. And then the testing will begin. The pre-test will start shortly, hopefully, and then the post-test, we should have everybody wrapped up, I hope, by, this is the government, so I have to say we expect. I can't commit to anything, but, November or so. At that point, we should really have a strong conclusion as to whether we're seeing something or not.
Smart Drug Smarts.
So a big thank you to Dr. Russell for taking the time to do that interview. Sneak preview about 10 or 11 months in advance. We will be having a follow-up episode at the conclusion of this year, as will be the end of the first phase of the SHARP program, and we will see exactly what they've learned, which of the interventions is showing the most promise, and where phase 2 of the research in the SHARP program is going to be going. So stay tuned for that. I also want to give a big hat tip to Rhiannon for arranging that interview and going through a few layers of bureaucracy to actually get that phone phone call that you just heard able to be made at all. This was not an easy interview to get, but I think a very, very worthwhile one. And I think it's really interesting the way that they're approaching this project, that it sounds like each of the teams that are trying sort of a battery of interventions on people, almost like the underlying assumption is that in order to increase fluid intelligence, it might require not just a particular chemical or a particular type of meditation or a particular electrical or magnetic stimulation to the brain, but a variety of these things working in concert. So obviously a ton of variables here, but in a way this is kind of like the dream study that all of us Smart Drug Smarts people want to see happening. So it's great that this is taking place. And now, as usual, the ruthless listener retention gimmick.
Smart Drug Smarts. Ruthless listener retention gimmick.
So nobody's really ever got anything bad to say about creativity. I'm just going to take it as a given that all of us would like to be more creative. And when people are testing creativity, typically what they'll do is give people a test like, here's a common object, come up with as many uses for it as you can within a fixed amount of time. And the more wacky ideas you can do with, say, a common stapler or something like that, the more creative that you're found to be. So it's long been sort of anecdotal folk wisdom that taking a walk can kind of stir your creative juices and make you more likely to stumble onto a great idea. You know, writers will be searching for the next chapter in the novel or a catchy headline or whatever, and they'll go for a walk and aha, they get it. But there wasn't really much scientific data to sort of back up this idea that walking spurs creativity until at the time, graduate student at Stanford named Marley Apezzo decided to actually get down and study this and see, is it the walking itself that is increasing creativity? Is creativity actually increasing? And if so, is it the physiological movement of walking or the fact that you're being exposed to novel environments as you walk around and that you're seeing more stuff than you would be if you're just, staring at your laptop? Therefore, it's the stimuli that are giving your brain more ideas to bounce around. So she got students, she put them on walking treadmills and gave them creativity tests that they could fill out while they were walking at a comfortable pace in a really sort of banal, boring room. So they're just staring at a gray wall, something like that. There's really nothing to see on this walk, but they're going through the physiological movements of walking. And lo and behold, the students who were doing the walking versus the ones who were doing the same tests while in a desk were significantly more, 60% more creative on the test that she gave them. That seemed like a pretty big result. So they did the test in a couple other ways. They also had students take a walk, then let them get off the treadmill, gave them a test a while longer to see if the creativity lasted or if it was only like a super acute response during the walking itself. And the creativity boost did stick with them for a while after the walk. And then they did similar tests with students that were walking around on the Stanford campus, being exposed to the sights, the sound, the smells, the whatever, which you might think would be a richer environment and spur even further creativity. What they found, interestingly, was it really seems to be the physiological movement of walking and not necessarily what you're walking around looking at that's the creativity boost. People did not have any significant difference in how much their creativity was amplified by walking around the Stanford campus versus just walking on a treadmill in a conspicuously boring room. Now, what this study doesn't answer is what's actually going on, and that opens the doors to a whole other bunch of studies, but apparently pretty legitimate science behind the idea that, just walking around a little bit can make us more creative thinkers for a while afterwards. So get your stroll on.
Smart Drug Smarts, the podcast so smart, we have smart in our title twice.
Okay, so you heard it. That is the episode. As usual, if you liked what you heard, please recommend this podcast to your friends through any and all means at your disposal. Word of mouth, social media, write smartdrugsmarts.com on a brick and throw it at somebody, whatever happens to strike your fancy. And also, next week's episode, in fact, it might even be like later this week because we definitely need to get this one out in advance of Valentine's Day. Sticking with our theme that we've done recently of trying to have holiday episodes, I'm not sure if we're going to do everything. I think we might have actually just missed Groundhog's Day. But our Valentine's Day episode is actually going to have viewer discretion advised. It's going to have some adult themes or not even themes. It's going to be talking about some pretty sexy stuff. So if you're under the age of consent or whatever, or blush easily, then you might just want to skip that episode. But on the other hand, if you like titillating stuff and you're looking for some interesting and scientific conversation to make around Valentine's Day, well then, do we have an episode in store for you? The show notes for this episode you just heard, of course, will be online at smartdrugsmarts.com, including the links to everything that I talked about here. And we'll be back at you very soon at a randomly determined time on the exact same podcast and with the same unflagging commitment to helping you fine-tune the performance of your own brain. Have a great week and stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only, although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.